effort on working on TB. And this work is just published in a paper in Journal of Immunology. And this picture was on the cover of that journal. And so the work was done with Selva Kumar Subian and Lambo Shi lab, and particularly Ranjit and Afzal, those two guys sitting right in the back. They did a lot of imaging work early on, and my contribution in this is creating images like this from their primary images. I'm going to talk today about these broad topics. We're going to talk about what we know about the functional organization of granuloma, and we will talk about roles of cells of innate immunity and adaptive immunity in controlling TB. We will talk about how mRNA expression can be used to define various cell types in granuloma tissue, and using single molecule FISH. And single molecule FISH is good for typical RNA, but for sparsely expressed RNA, we will use amplified FISH. And then we'll talk about detection of multiple RNAs in the same tissue. And then a little bit about these new platforms that have arrived that allow you to do image analysis of hundreds and thousands of RNAs at the same time, and how that technology or those technologies could be used to study objects like granuloma. Okay, so I will not talk about how prevalent TB is, how important it is. Only thing I would say is that TB has been with humanity since the beginning of the civilization or before. And so historically, TB was referred to as the decay disease or disease of wasting. You become very weak, and that was a telltale sign of TB. And for a long time, perhaps due to religious restrictions, dissection of human body was not allowed. And somehow in 17th century, people began to dissect the bodies, and they noticed that people with TB had these granule-like structures in their lung, and they were called tubers, and meaning swelling, and that's from that point on, people began to refer to the disease as tuberculosis. And first person to show that TB is caused by a bacteria conclusively was Robert Koch. He used to work in Berlin, and he's an amazingly brilliant scientist. He wrote a book. At those times, I think paper writing was not common. People just wrote pamphlets or books. And his book called Etiology of TB or Tuberculosis makes amazing reading. And I managed to get an English copy of that. And so he describes in this book how he was able to take inoculum from TB patients and in fact guinea pigs, and he was able to show that normally guinea pigs don't have much TB. Occasionally guinea pigs have TB in their studied cages, but when he was able to infect deliberately from these, actually he was, I think, one of the first people to do in vitro culture of TB bacteria, and also all his work was made possible because he was able to stain TB bacteria with trypan blue, and that you can sort of see in his. He made these drawings, and these drawings are amazingly they're telling you the organization of the tissue where the bacteria is found. And many of his drawings had this granuloma-like structures where this brilliant blue TB was present inside the granuloma. So from early on, since we know of the Mycobacterium tuberculosis bacteria, we know that it exists in these granuloma structures.
Is this a drawing he made from microscopic observation?
That's right. These are the drawings he made, and they exist. And I think there is a museum in Berlin where there's an institute with his name, and they have these pictures. I got them from a Wikipedia page. But he was, I think he had an amazing career. He went to Bombay to show that cholera was caused by a bacteria, and because there were a number of worldwide pandemics of cholera, and he was the first person to really show that it came from bacteria. Very interesting work. And so why is tuberculosis characterized by a granuloma? So granuloma has two main functions. One is to sort of surround and sequester the bacteria, don't allow it to disseminate throughout the lung tissue, because if it spreads, it would make the entire tissue sick. Other is perhaps equally important is to prevent the inflammatory response that the body mounts against the bacteria, to keep that also sequestered. So inflammation is mounted very vigorously, but it's localized to those sites. And I think these two key functions allow TB to survive in the body for a very long time. And most people will have TB sequestered in these granulomas all their lives, and they would never suffer from the disease. But some people do suffer, and the bacteria breaks through and cause system or whole lung wide infection. So a general organization of granuloma. So granuloma is largely a body formed by immune cells, and most of the cells in this granuloma are macrophages, and macrophages are the first cells that encounter TB, and they ingest bacteria, but bacteria mounts number of defenses against the cell that has ingested them. And the key one is that when the bacteria is ingested in a macrophage, it is present initially in a lysosome, and that lysosomes have a developmental path where they get acidified to destroy the bacteria, where the enzymes can lyse the cells. And TB manages to subvert that process and survive for long. But there is sort of tussle going on. When the macrophages get infected, they begin to secrete cytokines, and these cytokines attract other macrophages to the site, and that's the origin of this clustering phenomenon. It's like swarming of the cells at the site of infection, but additional morphological changes take place in macrophages. They become epithelial-like cells. They become like epithelium, and the characteristics of epithelial cells is that they sort of interdigitate, they form tight junctions with their neighbors, and that's what happens. So diagrammatically, the picture shows that the macrophages are sort of connected tightly to each other, and inside so because of this tight organization, many things happen. For example, vascularization of that area of the tissue becomes poor. Oxygen can't reach, the interior of the granuloma, and so it becomes hypoxic environment, and that hypoxia changes things, transcriptional regime pathway in the macrophages as well as in TB. And so that's one consequence. Other consequences that small molecules like drugs can have difficulty penetrating that structure. So the bacteria which is inside the structure is difficult to reach by the drugs. And that's experimentally this was shown by at PHRI by Veronique that drugs have difficulty reaching this site. In addition to macrophages, particularly at the periphery of the cell, there are cells of adaptive immune system, T cells and B cells, and other kinds of cells that that are going to mount a strong immune response. I'm mostly going to talk about macrophages and a little bit about T cells. And I guess I have talked a little bit about these issues already. Granulomas are very heterogeneous. They range from just a cluster of few cells to very large structures, few centimeters, even inches big, or taking a big portion of the, yeah. Yeah, that's right. A very important question. It turns out that this process that I just described, macrophages get infected, start secreting cytokines, attracting other macrophages, seems so fundamental. And this alone would allow the formation of the granuloma. And as a consequence, many other intracellular pathogens create granulomas. Not only that, even inanimate objects like particles of dust can create granulomas.
Only respiratory tract infections, or like those which happens in lungs, or anywhere?
No, I think it's broader than lungs, and I'm going to show you an example of that. Yeah.
I mean, you see granulomas with cystis on eggs, so you see it with extracellular eukaryotic thingies too.
Yeah, sure. So the key thing is that the begin as solid spheres, a cluster of cells. Then, in case of TB, they develop a core that is necrotic, where the cells die. And in case of active TB, this core becomes liquefied. There's too much cell mass, and that escapes. And individuals with latent TB will have granulomas that will have literally sterilized or force TB into dormancy for all your life. Lung may harbor different stages of granuloma, so it's a dynamic structure. We should not think of it as a endpoint. It continuously changes. And this figure taken from a very nice review in Cell by Flynn and Chan. Chan is not here, so if I make mistakes, there it is. Joe, if I make any mistakes about T cells, that's your fault. So a lot of this thing I'm going to describe comes from this review. And basically, the lung is organized into this air sacs or alveoli. Bacteria enters there and would be confronted by macrophages that ingest this, and they begin to move across the epithelium of these air spaces, and that is not a passive process because the epithelium is sort of closed, tight, and in structure, and that seems to be a actively controlled process. And it seems that different TB strains have different ability to permeate through the cell membrane, this epithelial layer. There are more infectious strains that Padmini studies, they may be able to enter through the layer more avidly than the other strains. But once it goes into the interstitium, that's where the granuloma would be formed. Attraction of the other macrophages will happen there. And importantly, a different kind of macrophage also would arrive. And these are bone marrow derived macrophages that come from the circulation. They will reach at the same site. And the transcriptional program of these two macrophages starts out to be different, but once they enter the granuloma and adjust with each other, their transcriptional program becomes similar. And I'm going to talk a little about T cells. T cells are really relevant for TB control. And the way the T cells work, their primary route of activation is that an infected cell, such as a macrophage, would pick up debris from the cells it has destroyed and actually present it on the surface of MHC2, which is always present, and there's also MHC1. But this presentation is made to a T cell that has a T cell receptor that comes in huge diversity, and just the right sequence, right kind of clone would be able to recognize this. And once that recognition happens, the additional interactions between this pair of cells that are important, and that allows activation of the T cell, and the T cell would begin to make cytokines. It will, and there are two kinds of T cells for this purpose. I'm just going to distinguish CD4 positive T cells and CD8 T cells. CD4 T cells will activate B cells and they have other functions, but CD8 cells will directly kill the bug by secreting all the cells that are infected with the bacteria, and they secrete things like enzymes, but together they control the infection. Important points about the role of T cells or adaptive immunity in TB control. So if you delete, for example, the gene for CD4 receptor in T cells in mice, or if you delete MHC, the mice becomes very, very susceptible to TB. And this is the work from Joe and Flint. Also in macaques, where CD4 depletion leads to very bad disease, HIV infection in macaques and humans makes the TB very exacerbated. The role of CD8 T cells is not well understood, and I'll come back to this a little bit. Even though these experiments that Joe and Flynn and others carried out showed that T cells must be relevant for the TB control, but how they do this is not really understood. Part of the reason is that there's something mysterious about the adaptive immunity in TB. For example, in macaque and also in mice, adaptive immunity kicks on a little bit late compared to other pathogens. Like in mice, I think it's 4 weeks, and it could be that antigen presentation that a viral infection makes, which immediately elicits T cell response, doesn't happen in TB. It could be that TB has a way to delay or otherwise perturb T cell response. In fact, there are a number of these theories that I'm listing here. The granuloma organization itself could be the source of delayed T cell response. So imagine that granuloma is hiding the the cells that initially confronted the TB, and that structure physically prevents the T cells to get in where they need to get in. And there's this sort of weird idea that I've seen in the reviews. Marilla would understand this, that the most dominant antigens against TB are those that are secreted by bacteria. And yet, if you try to vaccinate animals with those antigens, they yield very poor response. And we know this, that the BCG, which lacks those secreted antigens, is not a very good. So there's something weird about this T cell role in TB. And this next slide that I found in a very, very, very interesting set of papers by this group in NIH. They were able to in vivo image granuloma formation in mice. And the way they do this is that they're able to put essentially a camera right next to liver, and they study granuloma formation in liver rather than in lung. And liver is a special organ, so whenever you get infection in your blood, it immediately reaches the liver, because liver capillaries are sort of very, very porous. The cells are able to permeate them and enter, and the granuloma get formed. And so on what you see on the left is macrophages, tissue-resident macrophages in the liver. And in the liver, they're called Kupffer cells. And so they have formed a little granuloma and they stop moving. So these Kupffer cells were labeled with GFP. So they label this GFP in a different animal and took those labeled cells and adoptively transferred to the animal they observed, which was infected with TB. So this very clever experiment, they allowed to see the mobility of various cell types. So you can see macrophages stop moving, they form the granuloma, but T cells, and they were able to do a very sophisticated experiment where there are two kinds of T cells. The red T cells were specific to MTB antigens. They should recognize MTB, and the green ones were against albumin or something. So
Opposite. According to the antibodies, they are opposite.
I'm not quite sure, because P25, look, but big here.
Written is up. Okay, got it.
Oh, you mean the top label is bad?
Yeah. Just-
Oh, yeah, I'm sorry, that's the confusion. So P20, red is specific to TB, green is not, okay? And yet they are able to, these T cells are swarming around the granuloma that is not shown, dark area is granuloma, and they sort of go in and out, go around, but don't settle like macrophages, as if they're having issues permeating the body of the granuloma. And so these things tell you that there's something weird going on about T cell response in granuloma. I'm going to leave the literature here. Hopefully it's helpful to understand what I'm going to talk about.
Why they put the liver in fact? How common is dissemination of TB to the liver?
Probably not very common. So they actually inject TB into the bloodstream. And what happens is within minutes, literally minutes, it shows up in liver where it forms granulomas. So
It was hard to see the lung, right? Because the liver is not.
no, yeah, that's right. That's right. You probably can't image lung while keeping the animal healthy. But I think this has this unique feature that you can do this sort of initiate at time zero and observe rapidly. That kind of experiment is feasible here. But I think similar situation would, I mean, it's different than lung, but
I believe this is because of injected in blood. That's why.
Yeah. Yeah. So, with about in 2017, Lambo and and Selva came to me and said, can we image granulomas with single molecule fish? And so basically, TB, of course, should be studied in humans, but the tissue is not accessible. So people use, since very beginning, these animals, primates make granulomas and have a TB disease that's very close to human. Rabbits are models that mimic the human granuloma pretty well. They make necrotic granulomas. Guinea pigs, as you saw, Koch did work with guinea pigs, and they are very similar to rabbits. Mice is the most common host for all the TB work, but mice doesn't have necrotic granulomas. They make solid granulomas, but they don't necrotize, except in some special circumstances, such as in this mice strain that Igor Kramnik made. They make necrotic granulomas and few other typically known the mice strain that people use in the lab are highly interbred. And they don't make granulomas, but wild mice probably make necrotic granulomas. And then there's zebrafish. Maybe I'll say a couple of things about zebrafish. So a number of labs have done very beautiful work by making granulomas in the sides of zebrafish. And the zebrafish have both adaptive and innate immune system. What is special about zebrafish is that embryologically, the innate immune system is the first one to arrive, and the adaptive one appears late in the life. So you can make granulomas with just the innate system and ask how does the adaptive immune system interface with it. But it uses a different strain of TB, and of course, fish don't have lungs.
Then like bacteria, that's kind of TB. Like bacterial navigation.
Yeah, it's a different strain. It's not TB at all, you think?
Different species. Bacteria, so different species.
All right. So, but look, I don't quite have that nuance, but the people who do this, they treat that similar pathogenesis as TB. Okay. So since Selva had this model in rabbit, we begin to explore, can we use single molecule fish to study granuloma? So we have this technology of single molecule fish where we detect RNAs by a pool of about 50 probes, which are small oligonucleotides complementary to that RNA, bind side by side and label with just one dye. When you use this pool, you can deposit so much fluorescence on each molecule that it becomes fluorescent and is visible as a fine spot. So you see these kind of images in a simple fluorescent microscope. So in this case, this is for an RNA called CFOS, and which is normally not present in the HeLa cells, but if you induce them with serum, they will make this RNA very quickly, and you can see the RNA, and each spot comes from one molecule. In the same cells, you can look at some other RNA with a differently colored probes, and in this case, we did with Sijun, and you can computationally identify each spot and count it explicitly. So you can sort of create these kind of images where you know explicitly how many molecules of each RNA are present. So we thought we'll use this technique to look at granuloma in rabbit. So this is one of the first experiments we did, and we labeled, and all these experiments that I'm going to show you in rapid succession are in this paper, just published in Journal of Immunology. So here we imaged this granuloma. So one of the challenges of granuloma imaging with single molecule fish is that in single molecule fish, we use high power, high magnification objective to see each spot of the molecule. But if you want to image a big structure, which is about several millimeter or even bigger, you have to have low magnification to see overall organization. So in low magnification, you won't see the spots. And so that's a big conundrum. And second big issue is that lung tissue has a lot of autofluorescence. And this autofluorescence is dominant in green channels, but it's broad spectrum. It messes up even yellow and to some extent orange channels. So we can only image in red channels. So when we want to use the green channel, we assign them to an antibody. So in this case, we had an antibody against a macrophage marker called IBA1, and this IBA1 is expressed on the cell surface. So all the cells you see as green are macrophages, and all the red cells are making interferon gamma. So basically, you add your antibodies, you label them, and then you fix the tissue again, so that the antibody complex is fixed in tissue, and RNA is free to bind to the probes, and so this, yeah.
Why can't you red and far red?
Yeah, this is the kind of thing we do. We go far red, like size 7 as well. And so, in fact, some of the multicolor images were done with that way. Yeah.
This is in human, or this is in mice.
This is rabbit. So I'm going to show you some images with macaques, but mostly with rabbits. And so if you this is an enlargement of what I just showed you to show more clearly how the cells are distributed, and this is even more enlarged. And you can sort of see what I was expecting is spots of each RNA nicely detectable. That would be some evidence of specificity of the system. You can see some spots, but the tissue is one more thing to say about challenges of the granuloma imaging is for safety of these experiments, you fix the tissue in BSL-3 and fix it in high concentration of formaldehyde for weeks. And I think for the lack of convenience, for the reason of convenience, you keep it for months and years in that tissue thing. And so the tissue is highly cross-linked and messed up. And so in this, you can't see those fine grains, fine spots. But we did this for many RNAs. So this is a picture that Afzal took early on with IL-2, those spots you see are not molecules; they are cells, and clusters thereof. So this is an enlargement of that area, and green here is not IBA1. This is the autofluorescent cells. There are ways to quench autofluorescence, and we play with them, but in these images, it's present. Similarly, this is IL-10. I should say that cytokines are expressed when they're expressed at very high level. They become the dominant RNA in the cell, and so they're relatively simple to detect. And this seems to be present. So I'm going to just show you the granuloma. This is sort of unaffected tissue. That's the core of the granuloma. But within that, there are these clusters of IL-10 producing cells. This is a higher magnification. We did controls. So this is a control experiment where we made probes against GFP RNA, and you only see autofluorescence and no red. So that means the probes have to have an RNA to see. I'm going to transition to this little more complex experiment where we did multiplex labeling. in a solid granuloma. It's about 4 weeks early granuloma. And here the blue is interferon gamma. The colors have been changed. And the green is macrophage marker IBA1, and the red is TNF. And TNF is sort of central to macrophage activity. Let me not say more about TNF. What I'm going to show you is that this is sort of kind of images that we get. What do you do for this image? And what we did is we said, okay, we're going to analyze this. So that portion of that orange area, I'm showing a DAPI image. The DAPI was not present in the previous image. The 3 colors are those that are indicated. DAPI is missing, but DAPI was taken. And if you look at the DAPI image, there is this cool program, a machine learning program that is available now that can look at the sort of shapes of nuclei stained with DAPI and identify them even in the crowded milieu like this. And it sort of would draw a boundary exactly around the nucleus, and then you computationally say, okay, expand the boundary until you meet the neighboring cell's boundary. So we didn't stain the surface of the cells, but we reached some kind of this boundary was expanded until it met the boundary of that. So that's roughly the boundary of the cell. And with that, you can define exact location of cells in this complex tissue. And then you can ask, within that boundary, how much of other markers are expressed. And you can classify those cells. So in the same image, I'm plotting only those cells that were expressing IBA1 above a certain threshold. And in the next image, only those cells that express TNF and then interferon gamma. So for each cell, you can define whether it expresses interferon gamma or IBA1 or what combination of. Yeah.
This field has a certain depth, right?
Yeah, we treat this as flat. These are about 5 micrometer thick sections. And so occasionally there will be nuclei on top of each other. And to the extent they create errors. The errors are apparent in this image. For example, that cell was defined, all cells are defined by nuclei. So this cell is sort of capturing that area, but this cell is also getting a little bit of that signal. And here it's more apparent. So, it's not perfect, and this segmentation procedure to identify cells in complex tissue like this remains a challenge even today. So we did this, and so we found that most frequent cells, so there are about 900 cells that have only IBA1, about 450 have TNF, most cells have none, and very few cells have all 3 things being expressed. And what those cells are, we can guess, but having, yeah.
Did you look at the TAM interferon and IO10 co-expression?
No. So this combinatorial experiment, this is the only one. There's one more. Those become really interesting. I think we have to do all kind of combinatorial experiments. So back to that image. So once we know what cell is expressing which combination of markers, you can make these kind of maps where the color now indicates the expression of various markers. And the same color scheme, and you can sort of see the core of the granuloma has expression of multiple markers, and the periphery has just one or the other. Like, for example, this area has single cells expressing interferon gamma and so on. So what can you do with this? You can do something like, for example, a density measurement. you can ask, you know what cell types, in my case, cell types are those that are defined by this combinatorial expression. And we can say, what is the density of those cells that make all three things? And where is it most dense? And you find that white area is the one, the center, the core of granuloma is where cells are expressing all three markers. And this area has high expression of this interferon gamma marker and so on. So you can, this is just an illustration of what you can do with spatial information. And this is just the beginning, because many labs are finding mathematical algorithms to find characteristics that would be very interesting.
How do you find the threshold to see if a particular cell is expressing a marker?
Yeah, so you actually develop a histogram of all intensities for all cells, and then you can have a slider that says, okay, at this threshold, I consider it positive. And then obviously it's a sort of a little bit subjective. Yeah.
How do these images compare to images you get with cell-specific antibodies?
Say it again?
Cell-specific antibodies, would they give you similar images?
Yes, I'll come back to that question.
Are all granulomas behaving the same way?
Not quite. I'll come back to that issue also. In fact, this is comparing two kinds of granulomas. Looks complex, but it's very simple. The one on the right is a solid granuloma of the kind we just discussed. It has a solid core, necrosis has not appeared, and we're doing the same kind of analysis, but only two markers now, IBA1 and interferon gamma. If they express both, the cells will be yellow. If they express only IBA1, cells would be blue. And you can sort of see outside the granuloma, there's a preponderance of blue, and inside yellow. But those green boundaries are, again, the density measurements, where the high density of those double expressors are present. And you can sort of see in the solid early granuloma, there's high density in the core, but in the late granuloma, such as these, the core doesn't have those cells. In fact, it has no cells. You can't really find cells because cells have sort of died. You can't find nuclei and so on. But what you find is that granuloma is a late granuloma. It probably began in the core of the center, where the region which is acellular, and the bacteria was contained for a while, but then the secondary granulomas begin to form that look like fresh new granulomas. They have the same features. So this is quite often found in human disease, that there are these secondary granulomas around the primary one.
These green borders that you see?
Oh, so these green lines are simply a different way of drawing the density. So in this image, I drew the density by having the whole area green. In that one, only the periphery is drawn. This is the question of protein and RNA. Is RNA a good surrogate for protein or not? So we actually, this question keeps coming back after 3 decades of RT-PCR and 1 decade of microarrays and 2 decades of RNA-seq. So we should have sort of accepted that RNA is a good marker for protein, but there are cases such as this protein, HIF1 alpha. It's a protein that get expressed in TB in macrophages when there is hypoxic condition, and it's an unstable protein, and it's a core transcriptional factor that turns on a set of genes, but it becomes stabilized under hypoxic conditions. That's the idea. So question was, can we see RNA and protein at the same time? And be labeled with antibody and RNA, and you can generally see that very few cells have only RNA, most of them have both. At this point, I would like to show quickly that there were many RNAs that were not cytokines, that were expressed poorly. To detect those RNAs, we had to do something more sophisticated than single molecule FISH. And what we did was we used ampFISH, amplified FISH. And the way this technology works is that you have two probes that are not labeled by themselves, but these probes are designed like hairpin in the structures that's shown in the upper left. And when they bind to their target next to each other, they interact and free up this sequence, blue and green, that can initiate the chain reaction when you add these two labeled hairpins. And this reaction is called hybridization chain reaction. And these two labeled hairpins are sort of complementary to each other, but stay separate unless a initiator is present. So when you create the initiator by having a target, you can initiate this chain reaction that deposits hundreds of fluorescent dyes at the site of RNA. So it amplifies the signal, perhaps 55-fold or more. And to amplify the signal further, we have many pairs of these probes. I'm showing you 3 pairs bound in RNA. We actually use 20 pairs of these RNA, have these probes on the same RNA. So you should be able to get a very large signal for this. And we did this in macaque granuloma that we got from Robert Koch in Texas. Here we are imaging CD3 RNA, and CD3 is a marker for T cells. It's a receptor, therefore the RNAs are expressed at low level, and interferon gamma we have done. So we imaged with smFISH interferon gamma and with ampFISH CD3 RNA, and you can see both signals. And this is a sort of necrotic granuloma that you can see the core doesn't have many cells, and outside you have. And these images have the identified things, what cells we have identified by the color of the circle, and the red ones have only the CD3, green ones have only interferon gamma, and the yellow ones have both. So this is yellow. And so let me not blabber this. This is just a, but what you see is that there are very few cells that are interferon gamma positive and CD3 positive, those yellow ones. And that means activated T cells are few. And this is a result that is apparent from many other studies where people took granulomas, dissociated cells, and did flow cytometry and found very few activated T cells. And so, but you can sort of here, you can know where those cells are. One of the things that is still missing is we can still see about 3 or 4 RNAs, depending upon how many colors you have. So to sort of address this issue, I tried this technology that I adopted from others' work. And this is, I call this iterative FISH. And the way you do this is that, and I should take a moment to explain this because this would provide background for spatial transcriptomics. And what you do is you hybridize with a library of unlabeled probes for many RNAs. So in this case, let's say you have a library of 50 probes each for each RNA. So 50 probes for RNA1, 50 for 2, and so on. So same idea, the tiling of many probes on the same RNA. But the probes are not labeled, but they have these sequences that can be used to identify that probe with a secondary labeled probe. So you basically add these probes, you wait for whole night, you wash the excess of the probes, and then you add two label probes that will bind to the address tags of RNA1 and 2, and you will get a green and red signal, and then you erase those probes away, those secondary probes, and you remove your signal, and you come with a second set of detector probes that will bind to RNA2 and 3, and you can slowly build up an image for 8 RNAs. And I think we can do it for 30 RNAs. And what you have is, you have a dish which is open. And on the bottom of this dish is a cover slip. And the cover slip is called, your section lies on the cover slip, and you can add these reagents, for example, probes, or the dissociation reagents and so on, one by one, while imaging the section at the same time. The section doesn't move, and you can image again and again. And so this is sort of a simple experiment showing I did 8 RNAs, 4 rounds of probes, and this is a a stitching of the granuloma, and I'm showing you the round 3 for interferon gamma and ARG1. And so if you go from round 3 in that red area, you can sort of see some cells were visible in green, some little bit in the red, but when you raise that signal disappeared, and then you added 4 probes for the 4th round for the second set of targets, they reappeared. And so yeah.
What's the advantage of this method over what geomex and where you take a cell out and then you look at everything that the cell is expressing?
Yeah, I'm going to come back to that because I'm going to discuss the spatial transcriptomic methods right now. This is just a homebrew thing, a version of what people are doing on a larger scale. This is just to show that if you look at high power, that region of yellow, you can see 2 kinds of cells, some red, some green, some both, showing expression. You can also look at T bacteria by the auramine strain in part of this. And this is to show 4 rounds through, I think, the green square. But this is just the over lab's way of doing 8 RNAs, perhaps 30 RNAs. But look, the thing about this is that this technology has been automated by a number of companies. So I want to just take 4 or 5 minutes to describe to you how the spatial transcriptomic technologies work. So there are broadly two kinds of technologies out there: probe-based technologies and then sequencing-based technologies. And the probe-based technologies is OSMFISH, which is smFISH that I talked about, done repeatedly. So people have done this for 30 RNAs, but that's boring and it doesn't go far. It doesn't do thousands of RNAs. mudFISH and seqFISH, I'm going to say a little more in the next slide. And then there's this nanostring technology, which is also probe-based. I'll show a little bit more of that. And the 10x Xenium, which is older 10x genomics technology. Those groups are probe-based technologies, work slightly different kind of probes. They have high resolution, but low depth. The RNA, how many RNAs you can do is limited perhaps to 1,000 RNAs. Then there are the sequencing-based technologies, and they are characterized by 10x Xenium, SlideSeq version 2, and a very cool technology called StereoSeq from China. And they can do entire transcriptome by single cell analysis. And I will have just a little bit to say about each of these categories. So smFISH and smFISH looks complicated, but it's really simple. In fact, I talked about this about my homebrew test. So you do a combination of color coding and iterative hybridization to achieve hundreds of RNAs detection. And so if you can see reasonably that you can do 4 colors, and 4 colors provide you 10 possible combinations pairwise. So for example, you can have just single colors, top 4 rows, and then you can have red with green, red with yellow, red with purple, and then remaining combinations. So you can do 10 combinations with 4 colors pairwise, right? So you can have an entire library first hybridize and then detect first 10 RNAs and then come back with second set of 10 RNAs, and you can build an image for up to 150 RNAs. And this is what's shown in a paper by PHRI in, I think, 2015. And now there's a company which I just mentioned, Visgen does that, and Spatial Genomic smFISH does these exactly those that approach. And then there's this nanostring cosmics. This is most modern version of nanostring technology. It departs from old strings. There are no strings here, no nanostrings here. And the way it works is that you assemble a branch DNA amplified Christmas tree on your RNA. And so again, they have many of these probes bound to RNA long, and dozens of these will bind, and each will attract many reporters. The idea is to amplify, but do it specifically. You don't want to have wrong signals. And they had a compelling paper in Nature Biotechnology last year, and they were able to amplify and detect, I think, 1,000 different genes quite nicely in brain tissues. And they have a machine that you can buy and use. Just a little bit about technologies that are based on sequencing. So this figure is for slide seq2. And basically what you do is you have these beads, microbeads, and the size of the bead matters. Bigger it is, less resolution it will provide. Smaller it is, higher resolution it will provide. But these beads are laid on a slide, and location of each bead is known, and each bead has a oligo attached to it, which points to poly A of RNAs, but has a barcode, and the barcode is unique to that location, that XY location on the slide. And you sort of lay these beads on your slide, and you determine their locations in the barcode. And that the company will have done that. And so they'll provide you a slide, and then your tissue goes on top of it and sticks to it, and you have proteinase K that lyses the cells, RNA comes out and get captured on those here, oligos, and you do reverse transcription, and once you do reverse transcription, RNA sequence get linked to the barcode, and you can do deep sequencing, and you can go back and deconvolve the XY location from there. So these technologies, there are several kinds, but they're limited in the resolution of the bead. The Chinese company that I described to you has the best resolution, StereoSeq,
locations to within cell boundaries.
Yeah, so you also take an image of the section with DAPI or HNE, and since you know XY location of each bead and therefore barcode, you can link the two, and it's pretty compelling. All these technologies are limited by how good your segmentation algorithm is. Okay, just the last couple of slides. I'm not going to be here for long. So this is a poster by 10x Genomics, and they show the history of this very exciting field. And I want to point two things here. One is this. I can't read this. Let's see. So in situ hybridization was invented in 1969 by Joe Gall, and Joe Gall passed away this week, and I'll have a picture of him soon. And the second thing to point out is they were kind enough to cite Arjun's paper on single molecule fish as being the foundational thing for this. So this is single molecule fish, and then now it has proliferated into all kinds of technologies, including 10x own technology. And I will end at this, that this guy, he was 14 years old when he got a microscope as a gift, and he died, I think, 94. He would be. He had amazing career. If you want to know how they found that 2 helices of DNA run through chromosome, he did this. And it turns out Fred did those kind of experiments. Somehow he was connected with, he can tell how he was connected with Joe Gall. But they added, people talk about quantitative biology. He was adding DNAs, and looking under that kind of microscope and asking, how long does it take for the chromosome to fragment? And does it mean 1 piece of DNA running through it, or 2 or 4? And with this, I will end. And these are the people who did the work. Lamboshi, his postdoc, Chin Q, Selva, Subian, 2 postdocs with him, Ranjit and Afzal. Deepak Kaushal provided the samples from macaque, and Salvatore made many of these probes, or taught the kids how to make them. And Yuri Bushkin, who sort of wrangled me into TB world, and funding was these guys. Thank you.
Thank you. So beautiful presentation as always. I have a question that about the cytokine expression. Of course, I'm not gonna argue, the RNA versus protein, mRNA versus protein thing. I mean, I'm totally fine with that. However, with cytokines, we know that the cytokine expression is highly regulated, and also, for example, when we've done experiments, together, collaborative experiments on, for example, T-cell stimulation in vitro, we see that the release of cytokines does not correlate with the kinetics of the gene expression, and there are some cytokines that are expressed and peak at, say a few hours after stimulation, others that peak and remain high, others that they go down, etc. And of course, there is a whole complex regulation of cytokine expression, including the fact that when cytokines are made, they find a way to downregulate their own expression, not to go so that the cells don't go ballistic. So again, I know that it's not fair to ask, whether mRNA and proteins, but in the case of cytokines, there may be, and I don't know that there is anything you can do about it, but there may be a gap between the presence of cytokines in the microenvironment and the expression of the genes.
Yeah, Mahila, I think it must be strictly linked to the half-life of these RNAs. If they're short half-life, then the disconnect will become even more, right? So RNA will disappear, protein will survive for a while. And yeah, this is a complex issue for sure. And and that's why, you used to add brefaldin A in those experiments to make sure that the protein would not be released.
Right. Yeah, on the same line, I think on the same line as Marilla said, right? Then I had done a few years ago, I had done an experiment, CD34 cells I took, right, at the lab, and then differentiated them into myelin and erythroid lineages, and then looking for the differential expression of the genes, right? And then in that, one of the experiments, when we added the erythropoietin, EPO, to the CD34 cells, and then looked for the erythroid transcription factors, for example, GATA1, and then within an hour, you can see when we did the RT-PCR, we could detect the erythroid-specific transcription factors coming, expressing the RNA. And then when we did this simultaneously within 12 hours, and then we didn't see the protein. But the protein appeared late, after 24 hours or later, we could see the protein. So it was a very clear, when you start anything, when you induce the cells for any expression of certain genes, RNA comes first quickly, and then protein comes. And it makes sense that because from RNA, protein has to come, protein comes. So if you want to detect very quickly early what happens earlier, I think that RNA probes will be good. But if you want to detect, whether RNA probes or, antibodies, there is a question, right? So both are great, but if you want to do quick, early, immediately in an expression of the genes, if I want to do the timeline, I think the RNA is good. If you just want to do the qualitatively, and then count, I think the antibody staining is good. So that has been our experience.
Yeah, the advantage of RNA, of course, is that from genomics, you can directly go to probes.
Exactly, yeah.
And for proteins, you have to have an antibody. Somebody has to do the hard work of raising antibodies, showing that they're specific and so on.
Yeah.
But anyway, this is great. Thank you for staying through. And I had forgotten that there is an audience out there.
No problem. Great talk as always. Yes.
Oh, thank you.